back to Biostat Squid. In this tutorial we're going to cover SRA Objects, in particular the new SRA version 5. So if you're ready, let's dive in. SRA is an OR toolkit for single-cell genomics amongst other methods like spatial transcriptomics and ATAC-seq. You can read more about it in the Satya Lab main page. In this video, I'll be working with a single-cell RNA-seq dataset as a SRA object in OR. We'll cover the general structure of a SRA object as well as the standard preprocessing pipeline. So let's start with the structure of a SRA object. If we talk about single-cell RNA-seq data, we're essentially talking about a table of counts. The rows are genes and the columns are cells. We can then have metadata, which are annotations about the cells or the genes, and then we can have more complex data like reductions such as PCA, UMAP or TSNE, which reduce the complexity of our dataset to lower dimensions. We also have neighbors. And the counts can also have different transformations. Sometimes we need to work with row counts, normalized counts, scale counts. So all of these data types can be stored in a SRA object. The SRA object is a representation of single-cell expression data for OR. Each SRA object revolves around a set of cells and consists of one or more assay objects. The assays have single-cell level expression data, whether that is RNA-seq, ATAC-seq, protein expression. And if you're working with single-cell RNA-seq data, then the assay will be generally RNA, which is the assay we're going to be focusing on in this video. Each assay can have several layers. For example, when we first take our single-cell RNA-seq sequencing data and create our SRA object, we will create the layer counts in the assay RNA. Counts is the layer for raw data. But as you very well know, in single-cell RNA-seq analysis, we often use normalized counts or scale counts or batch-corrected counts to compare counts across cells, genes or samples or conditions. These different versions of the raw counts are stored in layers of our SRA object. I'll give you two examples. One of the key steps in data preprocessing involves normalizing our data. Normalization is the process of adjusting the raw read counts to account for differences in sequencing depth. That is the total number of reads per cell and other technical factors. And this ensures that the expression levels are comparable across all cells. This is important because in single-cell RNA-seq, different cells often have different numbers of reads due to various factors like the cell size, sequencing efficiency, or differences in the amount of RNA present in each cell. So, for example, if we wanted to compare the expression of gene B in cells 1 and 2, it would seem like the expression of gene B is 100 times higher in cell 2 compared to cell 1. But if we check the total number of reads, there were many more reads to begin with in cell 2. We need to correct for this, and that is what normalization does. The standard way is log2 normalization, which will make the reads comparable across cells. So now we see that the normalized counts for gene B are the same between cell 1 and cell 2. Of course, this is a very simple example with only 2 genes, but the point is that log2 normalization would make the counts comparable across cells and account for differences in library size. The normalized data in the SRA object is stored in the layer data. The second key step in data preprocessing is scaling. Scaling adjusts the expression values for each gene so that the expression values across cells are centered and comparable. It is particularly important in analyses like clustering or dimensionality reduction, where you want to ensure that all genes contribute equally to the analysis, rather than being biased by genes that with inherently higher expression levels. For each gene, We subtract the mean expression across all cells and divide by the standard deviation. This results in a scaled expression value that has a mean of 0 and a standard deviation of 1 across all cells. This process makes the data centered and scaled so that each gene will contribute equally to the analysis. The scaled data is stored in the layer scale.data. Great! If you're used to working with SRA objects, you might have already heard of SC-transform. SC-transform is an advanced normalization and transformation method specifically designed for single-cell RNA sequencing data. It is an alternative to traditional methods like log normalization and scaling. The SC-transform method uses a statistical model, in particular a regularized negative binomial regression model, to perform normalization, accounting for both technical noise and biological variation in a more sophisticated way than simpler normalization techniques. All this is to say that when we run SC-transform on our data, a new assay will be created, the SCT assay. And again, you can expect the layers counts, which has transformed or corrected row counts, layer data, which has the log normalized version of these corrected counts, and scale data, which has the Pearson residuals. If you're interested in reading more about how SC-transform actually works, you can check out my blog post on SC-transform. Nice! So what other information can we store in a SRA object? Of course, a very useful slot is the metadata slot. The metadata is used to store information about the cells, whether that is cell type, their percentage of mitochondrial counts, whether they were treated or not during the experiment. This enables us, for example, to subset the SRA object by a particular trait, or to remove cells with particular characteristics, or to do differential expression between cells of different cell types. The metadata is just a data frame with the cell IDs as rows and the different columns with metadata information. Easy. Now, while processing our single cell data, we often want to visualize our cells. Common dimensionality reduction techniques are PCA, UMAP, and TSNE. We'll talk about how to actually do this later on, but for now, know that all our dimensionality reduction results will be stored in the slot reductions. Examples are PCA, TSNE, or UMAP. Each reduction stores a dimensionality reduction object which contains information about the reduced coordinates, for example, UMAP coordinates, and additional metadata like the variance explained by principal components, for example. Finally, the graphs slot. It contains a list of graphs, usually a nearest neighbor graph, that are used for clustering and other analyses. The graph typically represents relationships between cells based on the gene expression similarity. For example, RNA_SNN represents the shared nearest neighbor graph for RNA-seq data. Or we also have SCT_SNN, which would be based on the SCT corrected counts. We'll talk a bit more about these graphs when we compute them. Finally, we'll quickly mention the slots commands and MISC, which stands for miscellaneous. Commands contains a record of the commands used to generate the object, and this can help in reproducibility by storing a log of the methods and operations that were applied to create or manipulate the SRA object. But in general, you don't really need it. And MISC for miscellaneous is used to store arbitrary information that doesn't really fit into the other slots. So it can be used to store additional analyses results or custom annotations. Okay, so now let's have a look at an actual SRA object and how a standard single-cell RNA-seq workflow would look like, as well as useful functions. Okay, let's have a look at an actual Seurat object and how standard single-cell RNA-seq workflow would look like, as well as some useful Seurat functions. So this script is for processing single-cell RNA-seq data using Seurat in R. It starts by cleaning up the R environment and loading the necessary libraries like Seurat and Seurat data, since we'll be working with a built-in dataset called PBMC3K, which is a classic dataset of peripheral blood mononuclear cells. So if you don't have the dataset installed, you can install it using this line of code. I already installed it, so I'm going to go straight into loading it. Okay, so once the data is loaded, we can take a quick look at the raw and unnormalized counts. Notice that when we first create a Seurat object, both the counts and the data layers have the same values. I believe this is so that the other Seurat functions work if they try to pull data from the data layer. But once we normalize the counts, these values in the data layer will be substituted by real normalized counts. Let's have a look at the metadata. The metadata slot refers to cell information. You can see each row has a cell ID. And for now, there's a few default columns. We have the ENCOUNT RNA, which is the number of reads in that cell, ENFeature RNA, which is the number of unique genes that were detected in that cell. And Seurat annotations are present in this classic dataset, but if you're using your own data, you will have to annotate your cells yourself. So this column here won't be present. Let's add some useful columns to the metadata. One of the first steps in a single-cell RNA-seq pipeline is to add some quality control metrics, specifically the percentage of mitochondrial and ribosomal gene content, which can tell us more about cell viability or potential technical noise. Okay, next comes the Seurat workflow. First, we normalize the data to adjust for things like sequencing depth or the size of a cell. Let's have a look now if our normalized gene counts have been updated with actual normalized values. So while processing our single-cell data, we often want to visualize our cells. Common dimensionality reduction techniques are PCA, UMAP, and TSNE. But before we do that, it is recommended that we identify the most variable genes in our dataset, which differ the most across cells, and they're the most likely to be biologically meaningful. Highly variable genes are the most informative genes, as they will help us identify different cell states or cell types. The function findVariableFeatures in the Seurat package aims to identify genes that are more variable across cells than would be expected by chance, and thus are likely to be biologically significant. This step just helps focus the analysis on genes that contribute the most to the observed heterogeneity between cells. I won't go into the details of it. You can read more about it at biostatistics.com, but there are different methods to calculate the variable genes. The most common and default one is VST, which uses raw counts from our RNA assay to compute neighbors, but you can also use SVP or DISP to find variable genes, which take the normalized counts from the data layer in the RNA assay. Okay, so when we run findVariableGenes, the function will store the variable genes in the gene metadata. Remember that anything having to do with genes or counts is stored in the slot assays. Similar to cell metadata, the gene metadata is essentially a data frame where each row has information about a gene. Since we ran findVariableGenes using the VST method, we have results for that method. Finding variable genes involves calculating the mean and variance of the expression of each gene. We can also see a column that contains true or false depending on if the gene was expressed or not, as well as the ranking. So how many variable genes do we have? By default, the find variable features will return the top 2000 most variable genes, but you can change this in the function itself if needed. You can also use the function variable feature plot to visualize the genes that were marked as highly variable. Great, so after that we scale the data so everything's on the same footing for downstream analysis. Scaled normalized counts are stored in the scale.data layer, as we already know. Let's just compare the raw, normalized and scaled counts for a few genes. Now, why is CD8A missing from our scale data? Looks like it's not a highly variable gene. Now, by default, scale data only scales the highly variable genes, if you previously ran find variable features. This is mostly for efficiency and noise reduction, so you're typically interested in the genes that show meaningful differences across cells. Scaling everything can slow things down and introduce unnecessary noise into downstream steps like PCA. So if you want to scale all genes, you can use all instead of HGV inside the scale data function. But it's generally recommended to only use highly variable genes for scaling and downstream analysis. Now, since we're talking about PCA, it's time to do some dimensionality reduction on our dataset. As explained in my previous videos, we can plot our cells based on the expression of two genes. For example, here we see cells that express high CD3D, a known T cell marker, and MS4A1, a known B cell marker. So we could suspect these cells over here being T cells and these cells being B cells. However, cell type identities rarely depend on just two genes. We need a way to account for the gene expression profile as a whole. That is why we use techniques like dimensionality reduction. Easy. PCA basically simplifies the dataset while keeping most of the important variation. Next, we can run PCA on our scale data, which will create the slot PCA inside reductions. The coordinates of the PCA scores for each cell are stored in cell embeddings, and loadings will be stored in feature.loadings. As you know, PCA reduces the dimensionality of our dataset, so choosing the top PCs will explain a good percentage of variance in our dataset. You can check out my other video on PCA if you're interested in knowing more. Now, the question is, how many components should we choose to include? 10? 20? 100? One way of doing this is by generating an elbow plot. It's basically a ranking of principal components based on the percentage of variance explained by each principal component. In this example, we can observe an elbow around PC 9 to 10, suggesting that the majority of true signal is captured in the first 10 principal components. Although in larger datasets, it's usually more like 50 or 100. You have more tips on how to identify the dimensionality of the dataset in the SRA webpage. And if you're using SCT transform, which we will cover in just a bit, the value doesn't really impact the results much. Okay, so next we'll compute nearest neighbours based on those top principal components and use it to cluster the cells. The find neighbours function in Seurat is used to compute the nearest neighbours of cells based on their gene expression profiles and stores the data in the graphs slot. RNA_NN stands for nearest neighbours. RNA_SNN stands for shared nearest neighbours. Let's have a look at the KNN and SNN graphs. So the KNN graph is just a matrix where every connection between cells is represented as a 1. This is called an unweighted graph. and it's the default in Seurat. We can have a look at how this would look like by plotting the first 200 cells. The black spots on the heat map represent ones. Those cells are connected, essentially. In the shared nearest neighbours graph, the connections between cells are weighed by how many neighbours they share. This is called a weighed graph. If we plot the heat map again, now we don't have discrete zeros and ones, but actually a scale of values from 0 to 1. In this case, 0 is white, so no connection at all. And then there's a color scale that goes through dark red to black, with black being the strongest connection. As you can see, the darkest points are in the diagonal, which is each cell with itself. Both weighed and unweighed graphs are suitable for clustering, but clustering on unweighed graphs is faster for large datasets, so more than 100,000 cells. Now, once the graphs are built, we can perform graph clustering. We've computed the distances between cells. Now we can decide which cells are part of a cluster. We can group or cluster the cells at different resolutions. The higher the resolution, the higher the number of clusters we get. Usually you should test different resolutions and see which one makes more sense for your data and your purposes. In Seurat, the function findClusters will do a graph-based clustering using Louvain algorithm by default. So that's algorithm set to 1. To use the Leiden algorithm, you need to set it to algorithm equals 4. See find clusters for additional options, but by default we will use the SNN graph we created in the previous step. Okay, so we've clustered our cells based on their overall gene expression profile. Now we want to visualize them in a plot. For that, we'll use a dimensionality reduction algorithm, which reduces the data to 2 dimensions we can plot. My algorithm of choice is UMAP, but I will run both UMAP and TSNE in this tutorial. We'll use UMAP and TSNE using the first 10 principal components. The data is stored in the reduction slot, and you can access the cell embeddings, which are UMAP coordinates, or just plot them with the function dimplot. We'll talk about some more visualizations at the end of this video. Okay, so this is an important step. Visualizing all cells in a plot like this one helps us see how well the clusters separate and whether the patterns make biological sense. Let's compare the different resolutions we computed. We see how the bigger the resolution, the more granularity we get. There's no right or wrong here. We would choose the resolution which best fits the cell populations you want to identify. For example, if you're looking to distinguish between myeloid cells, T cells and B cells, you might go with resolution 0.1 because it's enough to identify these higher level cell type annotations. If you'd like to distinguish CD14 positive from FCG or 3A positive monocytes, you might need a higher resolution that further divides this cluster 1 in resolution 0.1 into clusters 1 and 3 in resolution 0.25. We're going to cheat a little bit and use Seurat's annotations for this dataset, which, if you remember, are stored in the metadata column Seurat annotations. If we wanted to achieve similar level annotations in this dataset, so 9 different cell types, we would go with a resolution of something between 0.5, which has 8 clusters, and 1, which has 10 clusters. That is not to say that we might need a higher resolution if we wanted to distinguish between activated and naive CD8 positive T cells, for example, or a lower one if we didn't care too much about the subtypes of T cells. Okay, so if you're analysing your own data, you'll have to annotate the cells yourself. How do we do that? First, a useful Seurat function to know is dot plot and feature plot, which we can use to check for the expression of certain markers. You can see, for example, that cluster 1 using resolution 0.5 has a high expression of CD14, and in fact, they correspond to the CD14 positive monocytes. Another way of visualizing markers is directly on the UMAP plot, which you can do with feature plot. I won't go into the details of these functions, so check out the documentation if you'd like to know more. Now, checking for the expression of certain markers can be useful, but when you're trying to annotate your cluster of cells, it might make more sense to find markers for each cluster that distinguish it from the others. In other words, you're doing differential gene expression. There's different ways of doing this, and I encourage you to read Seurat's documentation, but I'll just run through the very basics of it here. If you're interested in a proper DGE analysis video, let me know in the comments below. Okay, so first we set the identity for the SRA object. This is the column of the metadata that contains the identity of your groups of interest. For example, if you wanted to compare B versus T cells, then we would use the column with the SRA annotations. But we're assuming that we don't have the annotations yet, so we will just decide on a resolution and compute markers for each of our clusters. I'm going to go with our lowest resolution, which identified 4 clusters for simplicity's sake. We can then compute markers for each cluster versus the rest with find all markers. The result is stored in DGE all, which is a data frame. We'll have a quick look at the top positive differentially expressed genes per cluster, so genes that are upregulated per cluster compared to the others. You can use these genes to then annotate your clusters. I'd also like to quickly mention that you can also compare one cluster versus another by using find markers. For example, if we're wondering what are the differences between clusters 0 and 2, we can use find markers to get the differentially expressed genes. The result is a similar data frame with the gene IDs, p-value, average log2-fold change, etc. The genes with higher positive log2-fold change are upregulated in cluster 0 versus cluster 2. The genes with more negative log2-fold change are downregulated in cluster 0 versus cluster 2. Or, seeing it the other way, they're upregulated in cells from cluster 2 compared to cells from 0. So obviously this is all very time consuming. There's many tools out there that have been developed for automatic cell type annotation, which help with this task. Some use markers, some use reference datasets that have already been annotated, and you can use those tools to get an automatic annotation of your cells. You can find some recommendations and tutorials in my webpage and channel. So imagine we've annotated our cell types and we have them in our column SRA annotations. It's always good to check if the annotations make sense by checking the expression of known markers for that cell type. And of course, you can also compare one cell type versus another to do differential gene expression analysis. So we've covered the main steps in a single cell pipeline. Great! If you've used Seurat before, you might have heard of SC transform. It's a specific type of normalization tailored to single cell RNA-seq data, which accounts for technical noise and variability across cells. This improves common downstream analyses like variable gene selection, dimensionality reduction, and differential gene expression. You can read more about it in the Seurat page. When we run SC transform on our SRA object, this will create a new assay named SCT by default with the transformed data. Counts are the corrected or transformed counts, data are the log normalized counts, corrected counts, and scale data has the Pearson residuals. Importantly, this is set as the default after running sctransform. So if you want to use your log normalized counts, you will have to specify assay RNA. Otherwise, Seurat will automatically use the sctransform data. Okay, so let's run sctransform. Note that this single command replaces normalize data, scale data, and find variable features. You don't need to run them if you use sctransform. During normalization, we can also remove confounding sources of variation, for example, mitochondrial mapping percentage. We then follow the same steps as before: compute PCA, find neighbors, and get our UMAP projections. Now, if we check the assays, we should have two of them, RNA and SCT. Each of them has different layers for raw, normalized, and scaled gene expression values. If you run SCT, it will be the default assay in your SRA object, so make sure you explicitly set the RNA assay as default if you need to use the original counts instead of the SCT corrected ones. Great, so before we wrap up, I should mention that if you're working with more than one sample, you will need to integrate them. SRA integration is a powerful framework designed to align and combine multiple single-cell datasets, such as those from different batches, conditions, technologies, or even species, into a shared space. It works by identifying anchors, so pairs of similar cells across datasets, using mutual nearest neighbors, and then uses these anchors to correct for batch effects and enable joint analyses. If you'd like to see a video on SRA integration, do let me know. But otherwise, we leave that story for another day. Squid-tastic! Thank you so much for watching. Hope this gave you a clearer overview of the SRA object and key functionalities, but do check out their website and documentation where you can find additional tutorials. But that's it from me. Have a squid-tastic day and see you in the next one.